Swayam Prabha. Digital India. Educated India.
Okay, so today's class is on inorganic chemistry in biology. Okay, what is inorganic chemistry? We have seen so far is the role of metal in different field. Let's say we have seen role of metal in periodic table, of course, they are full of them, and then we have seen extraction techniques. There again we have metal. Subsequently we have seen crystal field theory or the coordination chemistry chapter. Of course metal was there. In the last class we were discussing magnetochemistry, or that's the chapter, right? Or whatever the name of the chapter was. It's essentially magnetic chemistry, or magnetism we are trying to discuss. Now today we'll try to see if there is any role of metal in biological system or in us. Okay, it's a vast topic. It's literally impossible to do justice in one class. So we'll try to bring few topics, trying to keep it as brief as possible and try to convince you that there is an essential role of metal in our body or biological system or in nature in general. Okay, that's the roughly going to be the topic. So role of metal ions in biology. Okay, now I think discussing a little bit that nearly 50%, actually greater than 50% body weight is having metal, is due to the metal. Of course there must be some role. See, in nature the most important thing to understand is nothing is done in nature without a cause. We may not understand right now, or we may not discuss in the class, or maybe it's not understood by scientists yet, but nothing, absolutely nothing in our body is without a reason. That's, I think, definitely a big take home message. Okay, now of course metal complexes can act as drugs. The drugs we take, the drugs the doctors prescribe, a lot of them are having metal. Some of them may not be life saving, some of them it could be for flu, for fever, for indigestion, this, that. Some of them could be life saving, such as cancer, like advanced stage of cancer. There is a metal. Metal actually is saving human being from getting demolished or from getting dead or being killed in lot of cases. There are some examples. See, I am not expecting, again this chapter, I am not expecting each and every details of things you have to remember. I don't say so. I am saying that it is important to understand what is there in terms of medicine, in terms of biology. So there are, for example, there are some drugs, it's shown in here, which is operating on our different parts of body. For example, anything you can pick up. I think we will go for one of those, let's say platin, cisplatin you have heard of. It's a platinum containing drug. Carboplatin. These are specific drugs for some specific type of cancer. Of course, as you know, cancer truly has no 100% cure medicine so far. Different medicines are effective for cancer, for different type of cancer. Okay, so cisplatin, carboplatin happen to be two specific platinum containing drug which are effective for a particular type of cancer, and it saves a lot of people. Of course, it has side effect, lot of side effect, but nothing is more precious than life. So when there is no alternative, I think still better. Not every medicine has side effect. I mean, not every medicine has huge side effect. Almost every medicine has some side effect, for sure. But some of them has more, some of them have less. But essentially at the end, we end up having the best possible one at this given time. Okay, this is what is cisplatin all about. Cisplatin is a simple platinum containing molecule. So once again, we are trying to keep this class as simple as possible so that we can correlate, but there are more complex things which definitely you can understand, just we do not have time to discuss that much. It's a one class, we will try to give you an overview. So this is a cisplatin, this is the transplatin. Why it is cis? You can see that 2 chloro are cis, 2 amino groups are, or ammonia are cis. So this is cisplatin. Trans means they are trans to each other. Now transplatin is not effective as drug, only cisplatin is. That's mainly because the cisplatin, this amino group, let's say, which is labile, okay, I think let's say this is the amino group or whatever, there are these amino groups which are labile. Labile means they can fall off. Okay, they can fall off under a given condition. Let's say if you give a better ligand, metal is nothing but hold with the ligand. If you provide a better ligand, that better ligand can displace the existing ligand. It's like exchange, right? So what happens, this cisplatin can exchange, let's say 2 of its ligand, as is drawn in here, let's say 2 of these ligands, and it can coordinate. The better picture is not there. This question is also there in the tutorial part. We'll come and show you a little better picture, how it coordinates with the DNA. Okay, so the major problem of the cancer patients is, you cannot prevent the growth of the cancerous cell. Cancer is nothing but uncontrolled growth of your diseaseous cell or diseased cell. Right? That's why we see that swelling, right? Tumor. You see it's growing, there is no control. Okay? If something like, let's say, any part is having cancer, if it grows, of course, almost everything will get affected inside our body, right? So since the growth cannot be controlled, people are trying different way to stop the growth of the cancerous cell. One of the way to stop, the growth is to just cleave the DNA. You have DNA, all of you have studied, even if you do not at this point may not understand 100%, that's okay. So DNA is a base pair, right? It's a helical system. So this drug particularly can go and intercalate, or 2 of its ligand can be displaced by, let's say, 2 guanine. Guanine is there, adenine is there. So 2 base pair can displace, let's say, this ammonia. Definitely 2 of these ammonia can be displaced by those of the base pair of the DNA. Therefore, what happens is, once you have such displacement, you will not be, of course, there are more involved strategy how it exactly prevents. The moment such EG complex forms over there in DNA, you will not be able to carry out the cell growth. It gives a message, thereby, your cell growth process, you have heard of mitosis definitely, that overall process stops. Thereby, you are able to prevent the cell growth. Indirectly, you are killing the cancer or cancerous cell. If you do not allow cancerous cell to grow, that's it. Perhaps that's one of the cure of the cancer, right? And then, of course, you can do other treatment thereby. Once it binds, actually, then there is a mechanism called apoptosis by which it can kill the cell, even those cancerous cells. So what we have learned so far, a simple compound such as cisplatin, which is a platinum complex. You can write down the crystal field splitting and everything. Okay, it's a 4 ligand over there on platinum. 2 of the ligand can be displaced by the DNA base pair. You have heard of adenine, guanine, thymine, cytosine, uracil, and so on. Let's say guanine. 2 of the guanine from the nearby residue, let's say it's a big thing, right? DNA means it's a big thing. It's a long polymer type of thing. It's a peptide, right? From peptide backbone, from that protein backbone, you are taking two selective ligand. Those are ligand again, right? If you look at guanine, this is nothing but a ligand for metal. They can also undergo hydrogen bonding. That's how they are giving the helical structure and so on. Okay, but once again, they can be ligand. Ligand are nothing but having a heteroatom. What is heteroatom? Nitrogen, oxygen, sulfur. It has lone pair usually or a negative charge, and those negative charge is going to bind with the positively charged metal ion. Thereby, if you give an opportunity for all those ligands present in our body, some of them, not definitely all of them, some of them will bind with the metal and form metal complex. Okay, so that's what is happening in cisplatin. As I say, the base pair or this backbone, protein backbone, can bind with the platinum center, displacing two of the ammonia, let's say, and then we get a complex which will not allow the cancerous cell to grow. If you, let's say, break the DNA base pair, so that the region where the cisplatin binds, from there on, this DNA duplex will not exist, DNA structure will not exist. So the moment that collapses, lot of things changes, it triggers lot of things. So your mitosis or the cell division will not be progressing further. This is a simple way to explain. Of course, it is little more complex, but essentially that's how we see that it can be prevented. Okay? So I think you got an idea how simple complex can be effective as drug, as deadly as cancer, which are even very late stage of cancer can be cured by this drug. Of course, this drug has a lot of side effect, but still, I think when this cisplatin was not there, nearly 90 or 85% patient was dying. Okay, for now after cisplatin, so these are cisplatin and carboplatin. Carboplatin is specifically for prostate cancer. Okay, this disease now can be cured up to 95%, which is a lot. 85 to 90% people who are dying before this medicine. Now the people having this specific type of cancer where cisplatin is effective, nearly 90 to 95% people are surviving. Of course, it comes with a side effect, but still people are using it, and it's not that costly. Another thing is cost, how costly it is, right? Anyway, let's move on. But then essentially what this example shows you, that you have control on things. Cisplatin works, transplatin doesn't work. Okay? Therefore, you would be able to understand where to design your drug or how to design your drug. One component is which metal to choose. Okay? For example, let's say you take a drug with iron or copper. Iron center can get oxidized, iron 2 plus to iron 3 plus, and that's how we see the rust formation, right? Copper, copper 1 plus, copper 2 plus, this oxidation state occurs very quickly because that potential is achievable, right? Therefore, these are called redox active metal center, because you can switch between this oxidized state, reduced form and the oxidized form. Reduced form is for what? Iron 2 plus is reduced form, oxidized form is iron 3 plus. Copper 1 is reduced form, copper 1 plus, copper 2 plus is oxidized form. Now, these are the things you may not be able to use as drug, because let's say you use iron 2 plus. Iron 2 plus in air is also not stable. You put it in body, it may not be iron 2 plus, it got oxidized to iron 3 plus. Let's say iron 3 plus is not drug anymore, right? So you have problem. So you usually do not use some metal center which are having easily oxidizable or reducible center. Okay. Now these centers such as iron, copper, they can be electron carrier because they can travel between the two oxidation state very easily. Lot of reactions are happening in our body where we need electron. You have done the balancing of electrons, some equation A plus B goes to C plus D. You see that some electron is required somewhere. Oxygen plus 4 proton plus 4 electron going to water. That electron, where those electrons are coming from? Those are coming from some of these redox active site. These are nothing but enzyme. What is enzyme? I mean, let's loosely define enzyme. Enzyme is some center or some place in our body where a particular thing is going on. Let's say we are inhaling oxygen. We will discuss today. Oxygen is going to bind with something. That's the porphyrin center, hemoglobin you have heard of, right? That one will be calling metalloenzyme because it has metal in it. It's not necessary that every enzyme has to have metal in it, but those enzymes which are having metal, we will be calling it metalloenzyme. Enzymes are nothing but made of these peptides, lot of those amino acids. Okay, so we have heard of amino acid. One amino acid can be put together with another amino acid. Those are called amide bonding, right? So small amino acid put together, I have slide, I will discuss briefly. Put together will be a peptide backbone. Let's say we have how many? 23 amino acids, naturally occurring essential amino acids. Different sequence of these, you can put, let's say, different type, all these in different sequence. Let's say amino acid 1, amino acid 2, amino acid 3, then again 1, 2 and 3. This way different sequence you can give, amino acid will form peptide backbone. Those peptide backbone, okay, are the one can organize in different fashion. Alpha sit, beta sit, okay, all those you may have heard of, if not, don't worry. Then thereby will give you a big enzyme structure. So essentially if you look at enzyme and cut it down into pieces, what you get is amino acid. Different orientation of amino acid, there could be hydrogen bonding among them, there could be covalent bonding, they can form the amide bond, but enzyme is nothing but collection of amino acid in different sequence. I will be coming, okay. All right, so over here you have different way to control or design a drug. You can control the metal center, you can control the different ligand, what type of ligand you want, whether you want a high spin complex, whether you want a low spin complex, how the properties can differ. Even from high spin to low spin, this conversion is crucial for our existence. When we'll discuss the oxygen binding with the porphyrin iron center, hemoglobin, okay, will show how this high spin, low spin change is essential for our surviving. Without that, oxygen will not even bind, okay. So it all depends on coordination chemistry principles and the interaction of the compound with biomolecules or cells or tissue or organs, okay. Sometimes, amino acids are there and in between some metals are there. The role of metal could be different, okay. Sometimes what metal does is very simply, metal can hold different protein backbone together. How? Because metal can have ligand. The way metal stays in our body is either if nothing is there, water is there as a ligand. If water can be displaced with those amino acid backbone, amino acid has some ligand, let's say histidine, which is like a 5-membered ring with a nitrogen in it. So everywhere you will see whatever is binding with the metal, it has a heteroatom, nitrogen, oxygen, sulfur, this sort of thing are there. Either those lone pair or the negative charge is binding with the metal. So as I said, amino acid has these units where it can act as a ligand. These are monodentate ligand. It's not bidentate ligand. We have discussed bidentate ligand, right? Where you bind like a, bind very tightly. But the monodentate ligand may not bind tight, but if they are in the right place where the metal is there, metal binding site is there, if there is a space for metal to sit down, metal sits down there, those amino acid backbone can hold metal tightly enough to do lot of chemistry. Okay? It can act as drug, it can destroy a drug, it can create a lot of things. So this is what essential. So bioinorganic chemistry is nothing but structural and functional activities of the different metal ions. That's what we will try to discuss. You just read, I'll essentially discuss these in the class. Okay. Now, of course, every element in the periodic table is not readily available, you have learned. So therefore, what we see in our body is usually the one which is available in large quantity. Iron, for example, it is available in decent quantity. Bulk element like carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, these are present in our body in large amount in any biological system. Like organic molecules are made of these, carbon, hydrogen mostly, and then nitrogen, oxygen, sulfur. So those are bulk, but then there are trace elements, although very little present, but still they are there in our body. Iron, zinc, copper, and so on. Other different percentage of metal makes really our body. Overall, I think every time I see that there is a question, let's say give an example of trace element. This is one trace element which is essential for biological activity. Let's say over here, these are the one in the blue which are trace elements, but it is essential for biological activity. This is kind of periodic table with the biological perspective. You don't have to remember all these. One or two example if you remember, I think that's good enough. Trace element means small amount present, but those are going to be very important for us. Anyway, these are possibly essential. Okay, forget it. Let's move on. Now, what is essential element? I will not go through all these writing. That's for you. You can read. I will explain this. Okay, this chart. This chart is very simple. See, every metal is good, may not be for our body, for other purpose, but those metal which are good for our body is not good if you give a lot to our body. Let's say iron. If you keep on taking iron tablet or calcium tablet, like supplements you take, even over the counter you can get without prescription, do you think it's going to be good for your body? No. So there is always this curve. If you have a very less amount of some metal, then your survival is questioned. You may not survive. Okay. If, of course, you may be dead if you do not have a certain percentage of metal. Okay. If you do not have enough, then it is going to be deficient. You don't want to be deficient. You do not want to be super deficient. Then you will die. You want to stay in this region where it is optimal. Pretty understandable, right? If let's say 5 milligram is optimal for your body, you should have around 4 to 6 milligram. If you go 1 milligram range, you may be not surviving. But if 5 milligram is good, if you have 5 gram of it, you may be again going to here. Okay? So that is all this graph is going to tell you. It is going to be toxic up to certain level. After that, okay, we'll see you in the afterlife. Okay. Now, what determines which metal is going to be in our body or in biological system? Okay. What actually determines, what controls the selectivity? Let's say in porphyrin, that hemoglobin, there is iron, right? What controls these things? First criteria is definitely the metal has to be abundant, somewhat abundant on the earth crust. We should have some enough amount in nature for those metal to be here. Of course, every metal or every element that present on the earth may not be relevant in biological system, But whatever is in our biological system, in any biological system, those should be somewhat abundant. That's the natural abundance. Has to be somewhat there. It need not be the maximum, somewhat is there. In addition, it should be soluble. If something is not soluble, how can you really have in our bodies during our growth? We are eating food. So from food we get, let's say, iron, copper, lot of other metal. It should be soluble so that body can intake it, body can digest it, and it can be delivered. Okay. So essentially what happens is, let's say we are eating food. Food had some minerals, food had these metal center, different things. And then from our intestine, let's say it's a series of events. One, it's almost like a pizza delivery. Pizza is made somewhere, you place an order, okay. The delivery guy with a bike nowadays, okay, will come. Now it will deliver to a desired or to a order place, wherever the order has come from. So that's how it happens. So your food gets in, from there on whatever essential, lot of things are going on, and then there are different enzymes, and of course they are specific, they are very, very selective as well. Let's say iron is there, some guy is there, oh, okay, I have to deliver iron. He picks up iron, goes, and then dump it somewhere, right? They dump it somewhere. Then it could be further transported to some other place, or it could be stored. It's not necessarily everything we are eating is going to be digested or it's useful right away. It can be stored at a certain point, right? It can be stored or it can be delivered at a desired point. Okay. Anyway, so solubility is essential, oxidation state is essential. Oxidation state determines size. Okay. So let's say that delivery guy, if you give a huge pizza, it's not going to fit in his bucket, right? He is not going to deliver. So size is going to matter, ionic radius, ligating atoms. See, always no metal ions is free. Metal ions comes with a luggage of ligand. Now that delivery guy, if you do not give him enough money, he is not going to deliver, right? You have to pay him salary. Similarly, the metal ions, if they do not find the right ligand, they will not be ready to go, they will not be transported, they will not be storing, right. So all those things are essential to form or to do anything with the metal, and how they are delivered, how they are in the very first place, how they came, it depends on lot of factors. It's not just it has to be abundant. It has to be abundant. We have to have it on the earth crust. In addition to that, what is what is their characteristics, what they are made of, that determines. Let's say some zinc 2 plus, zinc 2 plus is not redox active, right? So if you have some active site means where the chemistry is happening, where you are doing some organic reaction. That is the active site. In our body, lots of active site is there, okay, where a particular reaction is going on, which is essential for our survival, right. Now, if you put something like zinc 2 plus in such site, where a redox activity is required, it's not going to do it, right. Zinc 2 plus, let's say, usually can do a structural job, means it doesn't change the oxidation state, it stays there, and thereby if different protein backbone has to be held together, zinc 2 plus is best. But then if you in that site, instead of zinc 2 plus, if you end up putting copper 1 plus or 2 plus or iron 2 plus, they can undergo oxidation reaction, they can undergo redox reaction. Therefore, your structural job, what you want, may not be done by them. You want somebody, spectator, some dumb guy to be there, like zinc 2 plus, which is feeling like a dumb, because they can do any chemistry, they cannot do any redox chemistry. You want somebody to hold the structure of the protein backbone You want somebody to be just there. See, you have heard of this Alzheimer's disease. A lot of movies been made. Okay. Parkinson disease, right? These are again very deadly diseases. Usually, not many medicines are there. It's painful diseases. Usually, what happens, let's say for Alzheimer's diseases, if somehow your body starts accumulating some metal center in an appropriate manner, in an inappropriate manner, means where it should not be there, it started concentrating. It could be at brain, it could be at lungs. Usually Alzheimer's disease, this could be at brain, let's say. So what happens? This, let's say you are storing copper 1+. Okay. Then those protein backbone start connecting with those copper 1+, let's say, and then it can agglomerate, it can precipitate out. You do not want that sort of things to happen, but it's happening. That's basically, let's say, Alzheimer's disease. In presence of a metal center, your protein backbone or proteins are getting kind of precipitated. If it is precipitating in some important place such as brain, your brain definitely getting affected, and lot of side effect, lot of things happens over the time, right? So that's how important the metal centers are. They are good chelator, or when they are the metal center, they can interact with lot of things, and thereby can change the biological activity. Okay, let's move on. Yeah. Sorry? Spin pairing stabilization. So, of course, high spin, low spin, let's say. Okay. So, sometime what happens, let's say if you have unpaired electrons, those unpaired electron means you are going to have a paramagnetic complex, right? Compared to that, if you have a paired, not in every situation, sometime it can be paired, if it is high spin to low spin, it can be paired, thereby those reactivity of those unpaired electrons can be somewhat minimized, right? Yeah, you can exchange the ligand, right? I am coming to that. How can you exchange the ligand? One, let's say water is there, you replace that with another nitrogen ligand, pyridine, let's say. You put pyridine instead of water. What happens? If it can change the spin state, then the chemistry will be completely different, right? Okay, so these are, all these points you may not have to memorize, but I think it is essential that at least few points you understand what exactly brings out. Okay.